So I can hardly see anything at all, but hopefully you can see something. And first, let me thank you for turning out this afternoon to hear a little bit about science, though I understand the festival was primarily literature for a long time. So I want to talk about the subject on the board. I don't know whether it's legible, but it says stem cell creation and prospects for cell replacement. I'm going to move these in case they interfere with the sound. So the bottom line of this talk is that all the cells we have in us are very stable. They don't change. But you can, by various manipulations that I'll talk about, arrange that they can. And so ultimately, we're in the business of trying to create new cells like eye, brain, heart from skin cells. That's the bottom message, and that's what I'm going to primarily talk about. So it says it'll go through matter of stem cells and then prospects for cell replacement. Now, this is not a Greek literature lesson, but I put this up because as long ago as 750 BC, there was writing by Hesiod which gave a remarkable insight to what we now know. For those who don't know their ancient Greek, I can tell you what a bit of it says. It says the long-winged bird rose up, but the liver grew indefinitely, and it grew every night by the amount that the long-winged bird, an eagle, had eaten that day. So it was an amazing story, and it relates to this picture here of Prometheus, who you can just about work out is somehow held there by chains. That's because Zeus decided that Prometheus should not have stolen the secret of fire from him, so he chained him to a rock. You can see the chain there, he's held down, and the bird, which is rather difficult to recognize, is there. That's its head eating his liver. And the story says that Prometheus's liver regrew every night by the amount that the eagle had eaten. Now that's amazingly, to an amazing extent, that's true. The liver can regenerate, doesn't quite do it overnight. But the striking thing is that when the liver regenerates, it puts back the amount that has gone. It's a rather amazing phenomenon. We know this is true. We don't still know why it's true. But that's a little piece of background history to this whole idea of being able to replace what is lost from our bodies. Now, in many cases, there is a natural process of regeneration, and you may know that the horns of a stag, this is a bit faint, they're up there, are shed each year, and they regrow. An enormous structure is regrown every year from cells in the head of the stag. So there are cases of regeneration, and some animals, like newts, can regenerate an arm if you cut it off. We ourselves have almost no capacity of regeneration, but the idea of replacement, replacing missing parts, goes back a long time. And this slide, this picture, shows the skull, the lower jaw of a Mayan, 1800 BC. And the fact is that two of the teeth are replacement. That one and that one are made of stone. Now that's not itself too surprising, because sometimes they used to prepare the deceased body for their assumed afterlife. But what is surprising is that the bone around these extra teeth has regenerated a bit. And so it means that when this was done, they had already got the idea of hammering stone teeth into the body while it was alive. Must have been pretty forbidding. But that goes back all that time. And then there is a somewhat imaginary painting here of Saints Cosmos and Damien. And this is the patient lying there, and he's lost a leg. Leg has been taken away. And they're putting another one on. Perhaps by good luck, they had it color-coded, and you can see that the hospital assistants had wings, angels, probably made it more likely to succeed. But in actual fact, that's an imaginary situation, and you can't actually just put another leg onto a human like that. So in effect, it's a kind of non-repeatable experiment. So this is the theme of what I'm going to be talking about. This is the ideal way things might work in future. So here's an elderly person, and sorry, the writing is rather blurred, but it says old skin. You take a piece of skin, and then you do this process called nuclear transfer, by which you create, by combination of a nucleus and an egg, an embryo. So the skin gives rise to an embryo, a ball of cells. These are called embryo stem cells, which I'll explain. And from these, when you've got that far, you can direct them into a whole range of things, including heart or brain. That's the direction in which things are moving. And if you do it this way, you get cell replacement with genetically compatible cells, meaning that you won't reject your own cells, even though they have now changed from skin to something which might be more useful. That's the overall idea of this talk. So I'll be talking about cloning and stem cells and prospects for cell replacement. So the first point that I've mentioned is that our cells are very stable. When a cell becomes brain or skin, it doesn't change. It'll go on many, many rounds of cell division and replication during our life, but it stays the same. However, this just illustrates that point. Here we start life as an embryo, and as the embryo cells go in the direction of, say, nerve, they can sometimes switch between brain and nerve, but they can never form liver or pancreas. Likewise, if the embryo cells have gone to be heart, they can sometimes switch between something nearby, but they can never become brain. Very great stability in our cells, and that's rather important. It's why we continue to live satisfactorily. Now, we now go back to the 1950s, and this was a major scientific question at that time, and that is, do all cells in the body have the same genes? You have to remember, half a century ago, we didn't actually know that. For all we knew, cells, as they form, could lose some of the necessary genes, and indeed, this rather fierce-looking German professor, Weissmann, concluded in the late 1800s that in order for our cells to form, we have to lose some of the genes for the bits we don't need. So if you're going to form skin, the idea was you would lose genes needed for brain or heart. That was the prevailing idea. To test that, we had to wait quite a long time for the following kind of experiment. In this case, one starts with skin, and the way it works is you have to separate the skin cells from each other, separate skin cells. And then you take the nucleus out of these cells, one of these cells, shown in a pinkish color, and take that into a microneedle so that the cell wall is broken, but the nucleus remains undamaged. Then you use this needle to inject that nucleus into an egg, and the egg's own chromosomes have been irradiated, And in some cases, this combination of the skin cell nucleus and the egg, unfertilized egg, can form an adult animal like a frog or a mouse. And we call that a cloned animal. To do this, you have to make special instruments, and you have to make a glass needle. When you break a piece of glass off, it usually has an irregular edge, and you have to make a special instrument which can turn that rough edge into this very sharp needle, rather like a wasp sting, so that it's so sharp that it can enter the egg without damaging the egg. So you have to make the instrument to get underway with the experiment. But when you've done that, it then becomes possible to make these successful cloned animals. And the first one of these was actually made in 1958. It happened to be a frog, because that's the kind of eggs which are very suitable for this work. And some people will tell you that cloned animals are in some way abnormal. This is by no means necessarily true, and this first cloned animal lived for about 20 years, which is a long time for a frog, and it had about 5,000 offspring, all of which were normal. So you can have very healthy, successful cloned animals. And as part of that technique, it was almost trivial scientifically to make a clone. In this case, the eggs of this dark-colored frog were used to receive nuclei from albino animals, and all of these frogs are identical twins because they're all derived from the same clonal experiment. In fact, you can graft skin between these and it's not rejected. That would be true of any identical twin. In this case, you can make as many as you want to make. If you wanted to make 1,000, you could. And these are identical twins, multiple identical twins, but that isn't really scientifically of great interest. It was a sort of consequence of the technique which attracted a lot of media coverage. More important was whether you can take part of such an animal, like the intestine, shown here, a formed functional organ, and take a nucleus out of one of those cells and do this cloning operation. And that was done, and in the course of time, one could make, this was all again done in frogs at that time, perfectly normal tadpoles, either from a fertilized egg or from the intestine cell. And when you look at the eye of the tadpole derived from the intestine cell, it's a perfectly normal eye. This is a lens and all the layers of the retina and so on. So it's a normal functional eye, but you have to remember that each of these thousands of cells was originally part of an intestine. So there's been a complete switch from the intestine to cells of the eye, and that's the general principle that this technique makes possible. You can do it in other combinations. You could take any kind of adult cell and in effect make any other kind of adult cell by this technique. So that shows that the stability which we're used to understanding for our own cells can be reversed, but it has to be done experimentally. In this case, using the normal eggs of the animal concerned. And in this case, we've grown those up. Here again, you can see that from the intestine, you've got muscle cells, perfectly good muscle cells. The green ones all came from the intestine, and the black ones in between are the animal's own cells, so that the intestine-derived muscle cells are just as good as the animal's own muscle cells. Then there was Dolly the sheep, which was a famous animal in, in the 1997, made by Wilmut and colleagues. And again, there was the common comment was that this sheep is not a normal sheep. Actually, the fact is that Dolly the sheep lived for about 6 years and was a perfectly normal sheep. It actually died when it was 6 years old, but the reason for the death was simply that it was such a valuable animal that they had to keep it indoors. And sheep don't like being kept indoors for all their life, and so they suffer respiratory disease, which happened to Dolly the sheep, and so it passed away after a very satisfactory 6 years. But there was, in fact, nothing wrong with Dolly the sheep. Again, cloned animals can be perfectly normal. So now I'm going to show a video of this technique. Actually, it's shown with mouse cells, and my friend Dr. Wakayama from Japan is the generally recognized most skillful person at this technique, and he lent me his video to show what it looks like when you do these experiments. Now here we see this is mouse eggs. You have to use a pipette to hold it in place, otherwise it skids around. That's the needle, and you pin the egg to the pipette so it's stable, and then this is a nucleus being taken up into the needle and now injected into the bottom of the egg. And then he shows it again in a moment. Here's another egg. There is a nucleus for another cell. Take it back a bit, and then in a moment you will see the nucleus being pushed right down to the bottom of the egg. You take it out again, and he does it once more, the same thing. So you can see what it's like. It looks rather trivial. Actually, you need very considerable skill to do this successfully, and he's the one that's been best at it. So it looks trivial, but actually it requires a lot of practice to make this, all aspects of that technique actually work. So that's the basic so-called cloning technique. So an interesting consequence of this experiment, now this is another one done by Wakayama, it's called serial nuclear transplantation, and it's very dim on the left side, but it's a mouse, and it says a cumulus cell, that's a cell from the mouse. So they take this cell, take a nucleus out of that cell, and inject it into the egg of another mouse. This egg is then grown to an adult mouse, another adult mouse. When they've done that, they take another cell from that mouse, again, same kind of cell called a cumulus cell, and transplants its nucleus into another egg and grow that to another mouse. That's why it's called serial nuclear transfer. Now, in this experiment, they did this for 25 times, one from the original mouse, each time growing the adult and doing the cloning experiment again. And in all, this involved, to get from the first one to the end, about 500 cell divisions. Now, that is a great deal more than any of our cells normally divide in our lifetimes. So this means you've extended the life of these cells beyond what is normal. And in fact, we could conclude that this is a kind of indefinite rejuvenation. Death doesn't seem to occur with this technique. Particularly interesting is the fact that there was no cancer. Normally, when our cells are divided a certain number of times, we are subject to cancer. But in this case, where you continually rejuvenate the nucleus through egg cytoplasm, you seem to avoid that. So it's a very interesting kind of experiment that we did a long time ago, but this one is really a more refined version of it. So now I'm going to turn to embryonic stem cells. And I suspect that most of you, if you were asked what a stem cell is, you'll probably say, oh yes, you've heard of that quite a lot. It's off frequently in the newspapers and so on. But nevertheless, not everybody really knows what a stem cell is. So let me explain that. Oh, I've got one more, sorry, I have one more video to show you. No, I've done that one. I'll come back to that in a moment. I'm going to talk about, first of all, about skin. Now, if you look at our skin, it's got the outer surface there. It's the bit of the skin that comes off all the time. And deeper down are these cells down there. Now, a small proportion of those cells contain skin stem cells. You can't say which one, because they all look about the same. But you actually know that one of many hundred have stem cell capacity. And I show this because these stem cells that exist in nearly all of our parts of our body are very difficult to identify, just like that. They look like any other cell, but when you test them, they are in fact stem cells. So the real discoverer of embryonic stem cells was Sir Martin Evans, who did this in the 1980s and quite appropriately got his Nobel Prize for that in some 5 to 10 years ago. But for reasons I don't understand, this has gone slightly out of order. I can't explain that. This is a slide I need for the moment. This is to tell you what a stem cell is like. Now, here is a cell, and this is the kind of cell in our skin, down the bottom, deep in the skin. When that cell divides, like all other cells, it forms two cells, two daughter cells. One of these daughters specializes and becomes a skin cell. That's why we call this a skin stem cell, because it can form a mature skin cell which will never divide again. On the other hand, the other daughter goes back into being a stem cell. So the stem cell makes two kinds of cells, one specialized product and the other another stem cell. That one forms the skin you think on your surface and doesn't divide, but this one, being a stem cell, will divide again. And when it does so, it forms two cells again, one of which becomes the mature specialized skin cell, doesn't divide. The other one goes back into being a stem cell. So when we talk about skin stem cells, these are the ones deep in the skin, which you can't easily identify, but they are ones which have this special property of making one functional product, the kind of skin you have, and the other one another stem cell. That's the principle of a stem cell. That's the skin again. Now, this is Martin Evans, who discovered the really important phenomenon of embryonic stem cells. Now, they differ from others in that they remain an embryo cell. That means that they can form everything. So when they divide, some of their products will go on and form heart and a brain and everything else. The other one remains an embryonic stem cell. And that was an absolutely crucial discovery because it showed that you can have permanent embryo stem cells. They remain all the time in an embryonic state. And that's absolutely central to the whole idea of cell replacement, because you have to produce those cells from skin or anything else to make them able to form every other kind of product. Now, this is a diagram to show the combination of these procedures. This was done some few years ago, taking a monkey, and actually, if a thing works with a monkey, it'll almost certainly work with a human if you were allowed to do these things. So they took the monkey, transplanted the nucleus, skin cell nucleus to the egg, as we've said, and you get an embryo. This embryo can now be some of those cells can be turned into embryonic stem cells, stem cell creation, and these are the cells, the Martin Evans cells, which will grow indefinitely. They grow forever, remembering that they are embryo stem cells with the full capacity to form every other kind of cell. And as an example, you can take these cells and you add what are called factors. They're actually gene products from other cells, which we know about, and you choose your factors carefully, and they can be turned into all sorts of things, including beating heart muscle. So I hope this will now show yes, there we are. These are beating heart cells, whole few thousand of them, cardiomyocytes, just the kind of cells you have in your heart that go on beating pretty much in time. And there's a whole sheet of them. Here's another picture, another group there and another group there. And remember that these beating heart cells are formed in the laboratory starting from adult skin. So the principle is that you can start with something like skin and create new beating heart cells. That's an amazing discovery. You'd think that might solve the problem of heart disease. It doesn't quite do so, because if you take these beating heart cells in the laboratory and transplant them into a living human, unfortunately the laboratory-based heart cells go on beating on their own timescale. They seem to think that they're still in the laboratory, and they don't synchronize with your own heart cells. That's a problem which exists but is gradually being cured. So eventually, this is how we think the rescue of some heart conditions can be obtained from things like skin. And remember, if these come from your own skin, you would have no rejection problems. So it's the principle that we're talking about. Now, we now move to the next stage, which are the so-called induced pluripotency cells. And this is work predominantly of Dr. Yamanaka, who received his Nobel Prize in 2012 for this work. And the key importance of this work is that he was able to use individual gene products added to embryo cells and convert them into whatever you might want, heart, eye, etc. Same principle as the cloning, but the real difference is you don't need to use eggs for this. Now, with the cloning procedure I mentioned, you have to use eggs, and it's extremely difficult, almost impossible, to get enough human eggs to do this work in a clinically useful way. Now, with his technique, you don't need eggs. You just take cells, and actually they can be from anywhere, add these particular genes to the cells, and they will go off and behave like the Martin Evans embryo stem cells. It works like this. You start with, say, a mouse, take cells, and I can hardly read it myself, but says you separate the skin cells, so they're loose cells, and you culture the cells in the laboratory, and you add genes. It gives them names, well-known genes. Add these genes to those cells, and occasionally, very occasionally, one of them will suddenly flip and become an embryonic stem cell. It's marked in red. And then you take that cell and grow that one up, so all the cells are now these new embryonic stem cells. And like I showed with the heart, you can turn those cells into brain cells, nerve cells. So you add factors and form the nerve cells, and then you can, in principle, transplant the nerve cell back to an animal which might need these cells. And this has been a huge advance, extends the field no end. And to summarize this advance, it says, conclusions from induced pluripotency stem cells. These are the embryonic stem cells. it says these can create embryonic stem cells and hence all other kinds of cells from skin cells, and critically without the use of eggs, because you couldn't get those in humans. It's also turned out to be extremely useful because you can make these cells from diseased patients, like Alzheimer patients or anybody else, and then once you have the cells growing in the laboratory, there's no reason why you shouldn't test every kind of drug you can think of. And companies will make you 100 million different chemical compounds. Of course, you couldn't test these on humans to see if they help diseases like Alzheimer's, but you can if the cells are growing in the laboratory and are not humans, just cells. So this is another great advance of this technique, and it's being applied very widely. So I will now take a moment for the experts just to summarize a really scientific question, and that is, when you've turned a skin cell into an embryonic stem cell, how does the embryonic stem cell decide what it should become? And this will take 2 or 3 minutes, and you can ignore it if it doesn't make sense. The fact is, there are only 2 ways this can happen. One is that when you have an egg, all eggs have different components at one end of the egg from the other. It's how eggs come. And when the egg divides to form an embryo of many cells, the cells at this end have the yellow component, the cells at the other end have the blue component. As soon as this difference has occurred, the blue ones signal to the yellow ones and change some of them into green ones. And this is the fundamental principle by which cells specialize. That's just how these things seem to work. I'm going to miss that bit out and come straight to the point what we call the genomic era. And this whole field really originated from Fred Sanger, who unfortunately died a couple of years ago in his 90s. He's the only one I know of alive who won 2 Nobel Prizes. Big enough to win 1, but into 2 is amazing. And he got the first one for discovering the structure of proteins consisting of amino acids, and the second one for working out the structure of DNA, or our genes. And this is now ballooned into an enormous field, the whole genomic area. As you know, for a moderate sum of money, you can find out what your genes are, which good ones you have, which bad ones. It all goes back, actually, to Fred Sanger. And one example of this very amazing genomic area is that when you have a pregnant lady, about 15% of the blood proteins in the pregnant lady's blood are actually those of the fetus, of the embryo she is carrying. It's now possible by this Sanger-related technique to sequence the DNA and so detect a lethal condition like cystic fibrosis at a stage early enough when it's still possible to opt for abortion. So if someone is unlucky enough to have a future offspring who is going to suffer something awful like cystic fibrosis or Huntington's disease, they can decide to terminate that pregnancy before the embryo has really started developing. So this is an enormously beneficial future direction of this genomic era. And this facility would apply very much to the kind of cloning or cell replacement procedure that we're talking about. Here is one example of how these things can be done. We start, this is to say, talking about the derivation of dopaminergic neurons, the problem with Parkinson's disease, from skin fibroblasts. So you take a mouse, grow some of the cells from the tail, Mouse doesn't really mind losing the tip of its tail. And these cells are called fibroblast cells. So you take those cells, and now you can obtain genes for whatever you want. So you overexpress the genes inducing these dopaminergic neurons into these cells. The changed cells with these genes multiply, and a couple of weeks later, you can see that the genes are functional. I have to read this here. And when the embryo is formed, there are these dopaminergic cells, and they can then be injected back into a mouse. And if you start with a mouse that suffers lack of these kind of brain cells, you can replace them in this way. So this is, again, the principle of combining these techniques of cloning, embryonic stem cells, genomic analysis for the relief of these serious disorders. So now we come to the question. I'm always asked this. People say that I have spent much of my life working with frogs, and indeed that's true, because they were the most suitable kind of species to use for exploratory experiments. When you want to see if something is going to work, you always try to do it with the most favorable material. And you may have seen frogs spawn in a pond. Each object you see, the frog embryo, is enormous by comparison with mice or humans. So, for example, each frog egg that we're accustomed to is 4,000 times bigger than a human egg. And that makes it much easier to do these rather delicate experiments. So one starts with that, and in time people find out how to do the same thing with the more difficult material of mice or humans. So that's why we did that. But people say, well, that's okay. Nobody really cares about a frog, nice as they are. Is it going to be any use to humans? And I think there's a rather immediate prospect in which this kind of field can be useful to humans. Now, I'm not reviewing my own work, but rather work that has derived from it. And I'm going to talk about age-related macular degeneration. And a number of people, older people, suffer from that. There's two sorts. There's the dry and the wet. The wet one means that you can alleviate it by putting in antibodies against vascular endothelial growth factor. And that is helpful, expensive, but it does help a bit. The so-called dry form is actually incurable. But there is a way around that now using this kind of technology. So here is a diagram of the human eye, or any kind of vertebrate eye. There's the lens, and light comes in, of course, from the right through the lens, and it impinges on the inside of the eye, and particularly in the inside of the eye are the photoreceptors, right down there. And they are immediately in contact with the retinal pigmented epithelium. This retinal pigmented epithelium has support cells which are necessary to keep the photoreceptors in good shape. Now, when someone suffers from age-related macular degeneration, the macula, or the middle of the eye, ceases to function, and you can't see straight ahead. If you look straight ahead, you just see a blur like that. To some extent, the peripheral photoreceptors still work. So you see a blur, but you can see something outside it. Eventually, even the peripheral photoreceptors die, and you can hardly see anything at all. So it's a very serious problem, affecting quite a substantial proportion of older people. Now, the way this works is the following. In your eye, you have the photoreceptor cells, and these cells have what are called rods and cones of these photoreceptor cells. these cells are in very intimate contact with the retinal pigmented epithelium. That's these cells there. They come right up against the photoreceptors, and they are needed to keep the photoreceptors in a functional state. Now, when you suffer from this macular degeneration, these retinal pigmented epithelium cells die. So there's one gone there, and the other one there. And when that happens, the photoreceptors also die. So there's now a gap there where the photoreceptors have died because the retinal pigmented epithelium cells that support them have themselves died. Now, an amazing technique is now functional from a scientific point of view. Here is the rods and cones of the photoreceptors, and you now see a complete new layer of replacement retinal pigmented epithelium. Here they all are in good shape, and they then connect up again with the photoreceptors and preserve them, stop them from deteriorating. Underneath are the deteriorated pigmented epithelium cells. They don't matter anymore, and the way this is done is to grow these retinal pigmented epithelium cells on a piece of plastic, quite a small piece of plastic, like this. It's about 3 by 6 millimeters, and I'm quoting the work of Professor Peter Coffey from London. Here is this sheet of plastic, and all these speckles are thousands of these retinal pigmented epithelium cells. They form a monolayer, a single sheet of cells, and they can be derived from skin or anything else, just like we've talked about. And this sheet of cells can be inserted surgically into the eye. It's actually not a very complicated operation. It's about the same as cataract. You can implant a new lens, you can implant this sheet of cells. It's the kind of thing that you would need to go to a suitable place, hospital, for a day. You don't even have to stay in overnight. And they can put this sheet of cells in there. And they look pretty good cells. Here they are as a monolayer. All these are the cells. So they can be inserted. And when you do that, you can at least rescue the deterioration. Your macular degeneration at least gets no worse, and in many cases it probably gets better. So this seems to be, in principle, a wonderful technique making use of cell replacement for the actual benefit of human health. So why is it not yet available? It is not available yet, and why not? One fact is that the cell numbers are important. Now, you all consist, as do I, of 10 to the power 13 cells. That's 10 million billion cells, some enormous number of cells, 10 to the power 13 cells for the whole body. Now, if you have a heart failure, you often lose about 10 to the 11 cells. That's 100 billion cells, huge number of cells. And you'd have to put back this number of cells to try and alleviate the heart condition. The amazing thing is that the retinal pigmented epithelium replacement requires a very small fraction of that number. It needs about 5 or 50,000 cells, which is a small number by cells. That's enough to rescue the failing vision. So that's why this technique seems to be about to be available for the eye, but not yet for the heart or brain. Now, there are other problems. What about ethical and legal issues? Now, I'm quoting Bob Edwards, who invented in vitro fertilization, and this is now an immensely successful technique. And the history of this is that when he first invented it, he had the most terrible press, and people would write to him and say, "This is completely unnatural. I hope you die in hell, burning alive," etc., etc. That went on for a while, and then as soon as it worked, Louise Brown was his first successful baby, the ethicists just vanished, went to ground, just like moths. They disappeared completely because it worked. So people have an idea about what is ethically acceptable until such a time as it works. I think the same is likely to happen with this whole field. So why can't we have this treatment now? The main problem is that the surgical treatment has to be approved by regulatory bodies. And these regulatory bodies are extremely conservative. And you can, in principle, bypass the regulatory bodies if you sign away your legal rights. But this is another problem with lawyers. They say they won't accept that unless you give informed consent. And they're likely to say, we don't think you understood well enough to give informed consent. So there's a blockage. Technique is available, works well, but cannot yet be given to patients. There are other legal issues, like what about the liability of the surgeon? Should the National Health pay? Are you allowed to pay privately? And finally, can the patient choose? I'm of the personally very strong opinion that the patient should choose, but not everybody agrees with that, and that's another problem why this is held up. Now, private health, which could deal with this quite well, operates in many countries, including China. So replacement hip or knee, $15,000, dental implant, $7,000, cataract, $6,000. The macular degeneration has cost about the same amount, $6,000. So some people could afford that. And remember that the cost of a blind person to the National Health is about $20,000 a year. So there's a very strong case for hoping that this wonderful procedure will be available, I would hope, as soon as possible. So now let me summarize this talk. What I've said, and this is the very first thing I talked about, all cells of the body have the same set of genes. Remember that if that were not so, you could not hope to take skin and make brain or eye or anything else from it. Luckily, all these cells have the same genes, and that was in fact the first purpose of the original cloning experiments. I then say that adult cells of one kind can be reprogrammed either through embryonic stem cells or directly, Yamanaka procedure, to cells of an unrelated kind, so skin to heart, etc. Now, the replacement of some kinds of aged or non-functional cells in humans is, in fact, a real prospect, and the problem is to try and persuade the regulatory bodies and even lawyers that they should allow this technique to be available to those of us who might need it. End of my talk. Thank you. Now, thank you. I understand that sometimes these talks invite questions, and I'm looking round because, happily for me, I have no problem with vision at all. I don't need any rescue there. But I have a hereditary weakness of hearing, such that if someone way over there asks a question, there's almost no chance of my hearing it. Now, this lady has very kindly said she's going to, if I can't hear the question, she's going to tell me what the question is. So shall we see if there are any questions, first of all? And I don't really have any more lights. It would be quite useful, as I can't see a thing, actually. The lights are beginning to... Here's a lady, yes, could you?
Yes, hearing problems. You have one.
Sorry, there should actually be one of these things you speak into, but there is one there. Sorry, yes.
Talking about hearing problems, you have a hearing problems. My
Yeah.
husband too. Is it any cure for that?
Can it be cured?
Yes.
Well, the long view is probably yes, but it takes a long time. There are the certain cells in the hearing which gradually cease to function, and it's a very complicated process to replace them, but that is one of the directions that's being followed, and I personally would very much hope it will succeed, but it's a very good question. So it's one of the more difficult ones. They call it cochlear implants, and you can grow these cells, these nerve cells needed in the ear, from skin. The big problem is, can you get them to take, as it were, to graft in successfully when you do the implants? That's in general the biggest problem, I would say, in this field, not making cells, but getting them to integrate. And it's very, very obvious when you come to the brain cells, Parkinson's and so on. You can actually get remarkably good brain cells in the laboratory which behave like the Parkinson's needed cells, but they won't integrate properly, very badly. And this is the big unknown, to try to find out how to make your wonderful cells working in the laboratory actually fit in to your own body. It's not a problem of rejection, it's a problem of somehow connecting up. Well, good question. Shall we, shall I try and take the question over there if I can hear it? Now, could you wait? Oh, there's one there. All right, let's take that one.
Hello, that was a very interesting talk, thank you. It's a bit of a long-winded introduction, but I'll get through it. I have a family with multiple health issues: diabetes, epilepsy, heart condition, rheumatological and neurological problems. So I have a very personal motivation for being interested in this field. And I missed the very beginning of your talk, so if you explained it then I'm sorry. But do you have a personal interest, and what is it that motivates you to pursue this field?
Yes, do I have a personal... Well, I mean, I would love to have my hearing better, but that's... if you reach my age and that's all you can complain about, then you're pretty good. So I don't... except for that, I'm lucky to be in reasonable shape. So, sorry, I will have to repeat that. But, yeah.
So if not, what's motivated you to continue against a lot of ethical criticism? What's motivated you to continue in your field?
Oh, I see. Yes. Why do I continue working? Yes. Yes. Well, that's right. Why do I go on doing, which I do? I show up for work quite early in the morning, and I work full time, in spite of my age. So the question I'm concerned with is, what is the mechanism by which an egg can rejuvenate an adult nucleus? We know it can, like I've explained, but we have absolutely no idea how it does it. Now, you might say, well, why bother? The fact is it works anyway. But the experience is that if you understand the mechanism, molecular mechanism behind something that works, it becomes enormously more useful. And the Yamanaka procedure, which is widely used, works at an extremely low level, like 1 in 1,000 cells will make this switch that you need. And that's a problem, because if you're trying to grow 100 billion cells, you have to grow that cell for an enormous length of time in the laboratory. And when you do that, it tends to go wrong eventually. Something else happens wrong with it. So if we could make the replacement procedure for these cells a great deal more efficient, that would help enormously. And the position is that the egg has that ability in a way that other techniques don't. So I believe there's a future in trying to understand how the original cloning experiments actually work. So that's my personal motivation for keeping on working. Now, I can't see very well. Do you want to try a gentle... Or anyway, you choose, please, because I can't even see what's going on.
Over here.
Where are you?
I'm here.
Okay.
Here.
Okay, thank you. I can see someone waving.
No, it's here.
Over there. Well,
you showed a really interesting film of heart cells beating in the laboratory.
yes.
What I was curious to know is what determines the rate that they beat in the laboratory? Because you said that there was sometimes, quite often, a mismatch between the patient's heartbeat and the laboratory heartbeat.
Yes, so why don't they...
What determines the rate at which the ones in the laboratory are beating?
Oh, I see. That is probably the so-called pacemaker. And you can get replacement pacemakers. Now, a group of cells like that will have pacemakers in them, but my understanding is that these pacemaker cells are not identified. So you don't know, when you have a group of cells like that, whether it's one pacemaker or two or none. And the problem is to understand the pacemaking function of this group of cells, most of which are cardiomyocytes, so that can be controlled. Now, this is a particular problem of the heart, And if you do, for example, grafting skeletal muscle into a body, a person, that integrates reasonably well. It's a special problem of the electrical properties of the heart. And I have no doubt, so many people working on this, it must eventually be solved. But at the moment, that is a substantial hold-up, probably due to pacemaker cells, and we don't know enough about them.
How impeded...
Is that the question?
Yes.
Yes, sorry.
How impeded has your research and your colleagues' research been by those pesky regulators and lawyers?
Oh, how inhibited has it been? Yes, well, in my particular case, I have to confess that it has not, because the regulatory bodies and lawyers are very uninterested in frogs. Makes it very convenient for me. So that's a sort of bonus of working in an exotic animal. But it is proving quite troublesome when you get to doing these things anywhere near humans. And I really don't understand it, because, for example, if some company developed a drug which would benefit 999,000 people, but it might kill one person, they say, okay, no good, unacceptable. And if anyone uses it, we'll impose a massive punishment of billions of dollars. Now, I think they ought to be more quantitative and say, if you prevent drugs being used because they were, in their judgment, they were not fully tested, you are damaging the 999,000 people who would benefit. So there should be a sort of balance between benefit and potential harm. And I suppose if you tested enough million people, you'd find someone who will die of drinking coffee. But that's not enough to eliminate the use of coffee. So this is a personal view. Answer your question, as long as you work with animals, you're not troubled so much, but it really is a problem once things have to be validated for human use. No, I don't know. I can't. Well, shall we take this gentleman? He's got a prominent hand up. I'm just going to keep an eye on it because some people have to go to another talk.
Right at the start of your talk, you talked about newts and the way they grow things. Is that of any benefit in what you're doing? Because you were talking about injecting cells into people. Can you apply the right electrical stimulus or whatever to make these things magically grow?
Yes. People have been trying for a very long time, and nobody absolutely understands why it is that a newt, if you cut its arm off, at this point it'll make a complete new arm and elbow. If you cut it off that way, it replaces what's missing. And it's, I think I have to say, completely obscure why that happens. And for example, it doesn't happen in frogs, or for that matter in humans. It's a very peculiar characteristic of newts. And people would love to know why, and they just don't. The one thing I would say about that is that when you cut the whole of an arm off a newt, it makes another one. It takes about 2 months to do it. There's no reason to think that the cell types actually change. So the blastema, as it's called, the lump when you cut it off, has cells which have despecialized from bone and skin and muscle. They look like neutral cells. But when you grow it, the ones that were muscle go back into being muscle, and the ones that were bone go back into being a bone. That isn't quite equivalent to what we're interested in doing, is making skin form something completely different. So desirable as it is, of course it is, to know how a newt does it, it might not be all that much use in the end. Well, I can see a hand up here. Would you like to bring there's a loudspeaker thing there, yes.
To what extent does your research relate to cancer research and vice versa?
Well, it's, I think one have to say rather indirectly, the major problem of cancer is the continuing proliferation. So when you have a cancer, the cancer cells don't divide very fast. It's not a rate of division, it's the continuing division. So the cells divide, and then they wait a bit and divide again. And they never stop dividing. So that's a problem of proliferation or continuing cell division. And our work is really more concerned, in this whole field, of switching cells from one kind of specialization, like skin, into another, like brain. It's sort of, it's in the end, these things all connect up, but I would have to say I think this is rather indirectly related to the problem of cancer. Someone must say when you need to, I think it's an hour already, isn't it? We've been going for about an hour, so when do we need to stop? Well, this has got...
5 minutes.
Sorry?
We've got another 5 minutes.
5 minutes, all right. Well, I mean, some of you need to get to some other lecture, so I mustn't delay. In fact, walk out as soon as you're bored. But meantime, I can see a hand up over there. All right, well, let's take... Gentlemen.
I don't have a question. I just, having worked with you for 25 years in cancer, I never failed to be astonished by the clarity of your presentation, but above all, the modesty.
How kind.
What I'd just like the audience to know...
On that comment, I feel rather like Winston Churchill, who said of Attlee, he's got so much to be modest about.
The audience should know that this is the stem cell of all of that research. He's unbelievably modest. He did all the first experiments, and he is the man for whom we should all be grateful, including all these other authors whom he very generously quoted. He didn't once say, I did this, but he did. So well done.
Well, thank you very much. Thank you very much. Yeah, thank you everybody very much. Thank you. How kind. Very generous comment. Now, do we have any... I think there's about 2 minutes left, so someone should take the loud... one over there, is it? Yes. Okay, yes. Thank you.
Is there any prospect from this line of experiment in reproducing human eggs themselves with a view to use in fertility treatment, for example?
Reproducing human eggs from cells, yes.
So that egg donation would
Yes, it's a very good question.
DNA
that's a very good question and a very relevant one. There have been quite a few efforts in that direction. And people have got quite far. They can take these Evans embryonic stem cells and get them to enlarge, and they look rather like eggs. But as far as I know so far, those eggs do not fertilize. So they look pretty good, but they don't actually work. And maybe in time they will. But of course the worry is if they don't form functional eggs, you're not sure that you can use them very effectively. But it's certainly a direction people are going in, and it's still, I would say, very obscure why a few of these cells suddenly decide to start becoming egg-like. That's very, all this is unclear. And the world as a whole has thousands of stem cell institutes springing up all over the place. And some of these will be concentrating on that area. But it is surprising when I go to other countries all over the place, they always say, let me show you our new stem cell institutes. It's a massive kind of tidal wave of activity. And all these people working on it, I feel almost sure these problems we've mentioned will eventually be solved, as long as we don't have too much interference from regulatory bodies and lawyers. I think we should discontinue, and thank you very much for your attention.